Hi, welcome to this week's episode of Science for the Non-Scientist. I'm Shamim Sinar. This week, I'm going to talk about this really cool paper that explores how bacteria in our gut can protect us from viruses. So our immune system is pretty cool. Not only does it fight infections once they've started in our bodies, but the immune system can actually prevent infections from starting in the first place. And one way it does this is by creating this antiviral state in our bodies. Basically, it releases all of these chemicals that make it really difficult for viruses to gain a foothold in our bodies and to grow and to replicate. So exactly how these bacteria in our gut help create this antiviral state is explored by these researchers from Harvard, Yale, and the Howard Hughes Medical Institute in this recent issue of Cell. So these investigators then look at lymph nodes in the mouse intestine. And like mice, we also have lymph nodes in our intestine in this tissue called the mesentery. And the role of these lymph nodes is to basically be on high alert for any bacteria or viruses that might want to start an infection. And so the researchers look at a set of immune genes and measure the activity of these genes in two different kinds of mice, okay? So the first kind are wild type specific pathogen free mice. This means that they don't have any genetic mutations that we know of, and they don't have any infections that we know of. Basically, they're healthy mice. And then they look at mice that have been treated for 7 days with antibiotics in their drinking water. And this basically kills the gut bacteria of these mice. And what they find is that the healthy mice have pretty high immune gene activity. Okay, you can see on the scale that bright red here means pretty high immune gene activity, and bright green on the other side means pretty low gene activity. On the other hand, the mice that have been treated with antibiotics have really low gene activity, right? In contrast to the healthy mice. So what this indicates is that killing the gut bacteria turns down the immune activity in these mice. And as you know, in the mice and as with us, it makes us more susceptible to viruses. So one might say, well, how do you know it's the bacteria that are responsible, right? Maybe the antibiotics are doing something else, and it's some other function of the antibiotics that's causing all this immune gene changes. So what these researchers did is they looked at a specific kind of mouse called a germ-free mouse, okay? And these mice are basically raised to be free of any kinds of germs. They don't have bacteria inside of their bodies. They don't have bacteria on their skin. They don't have any bacteria anywhere. And so they looked at these mice and looked at the immune gene activity in these mice, okay, of these genes. And what they found is that for most of these genes, the activity in these germ-free mice in the light gray was lower than that of the normal mice in the black. All right. So this is pretty important. What it shows is that those germs are necessary for optimal immune system function. When these mice don't have these bacteria, their immune system doesn't work as well as a healthy normal mice. All right, so then the researchers think, okay, well, what's controlling the activity of these immune genes? And we know that one really important protein in our body that controls a lot of aspects of the immune system is called interferon beta. And so they ask, well, does interferon beta have anything to do with all of this? And what they do is they look at mice that don't have any interferon beta. Okay, they have a genetic mutation, so they don't make any interferon beta. And what they see is that the immune gene activity in these mice without any interferon beta is also much lower compared to the healthy wild type mice. All right, so this is basically the model then. These bacteria are somehow stimulating our bodies to make interferon beta, which then helps to turn on all of these immune genes, okay? Now, when you give antibiotics to these mice, it kills the bacteria, and so the bacteria can't stimulate our bodies to make interferon beta, and so we don't have good immune gene activity. Now, if you don't put any antibiotics in the system, but you take a mouse that doesn't have any interferon beta, you're going to get the same downstream result, okay? These immune genes are not going to be able to work well because you've got this interferon beta missing, all right? So the next thing the researchers do is to say, all right, well, we've shown that these bacteria are important. Can we identify a specific kind of bacteria that's important for this interferon beta activity? And so they look at Bacteroides fragilis, which is one of the most common bacteria in our gut. And here's an image of them. They're stained pink in this image so that you can see them under the microscope. And what they look at is the interferon beta activity in regular mice and in mice that have been injected with a protein from this bacteria, Bacteroides fragilis. All right, and so this is the normal level of interferon activity in regular normal mice. But when you inject these mice with a protein from Bacteroides fragilis, you can see that the interferon beta levels just skyrocket, okay? So this shows that it's something about the protein from this bacteria that's stimulating interferon beta production in these mice. So at this point, they say, okay, we know Bacteroides fragilis is one of the bacteria that's really important to increasing interferon beta activity, all right? They say, well, can we actually show that increasing interferon beta activity helps protect the mouse against viral infection, or at least makes the infection less severe? And so what they do is they take these mice and they inject them with a virus called VSV, which is vesicular stomatitis virus. And this virus can make the mice sick. And so they look at how sick these mice get, how sick normal mice get, okay, when you inject them with the virus. Then they see, well, if you wipe out their gut bacteria with antibiotics, how sick do they get? All right, and you can see that they're much more sick. This is the cumulative disease score here. So the higher the number, the more sick the mouse. All right, so treating them with antibiotics and killing off their gut bacteria makes them much more sick. And that's shown here. If you don't have bacteria, they're not able to stimulate interferon beta production as effectively, and their immune gene activity is not as high. Now, interestingly, if you wipe their gut bacteria out with antibiotics, but then you give them PSA, which is that protein from the Bacteroides fragilis, right? Then you can prevent them from getting as sick, all right? They're no sicker than the regular mice. Now, it's a different story if you use mice that don't have any interferon beta at all. Here, what happens is that because there's no interferon beta, they can't make immune genes, even if they have normal bacteria in their gut. And so all of these mice, whether you give them antibiotics, whether you give them the protein back, whatever, they're all pretty sick. All right, so the main message then of this paper is that killing the gut bacteria results in less immune gene activity, and this makes the mice and us more susceptible to viruses. Thank you for joining me for Science for the Non-Scientist this week. I've got new episodes every Friday. Please subscribe.